Well, thank you, everybody, for popping on. I think that's probably enough time. It seems like the participant addition has slowed down a little bit, and so I'm super excited today to hear from Zeiss. And so we'll be hearing about the improvements that they've made in terms of both getting the Lattice Lightsheet commercialized, which I think is a tremendous feat, and it is really impressive. And I'm super excited to I got a sneak peek a couple of weeks ago to hear where they're coming from at this, and I think everybody's going to get a lot out of it. I'm very excited with the direction that today's going to go. I'm going to turn it over to either Monica or Kevin, whoever would like to kind of actually do a formal introduction of the team and who will all be presenting.
Hi, thank you, Brandon. I just wanted to thank everyone here for inviting Zeiss to be a part of this event and be a part of your users group. We're here today from Zeiss to not only discuss what we've implemented so far to help streamline the data handling and analysis of Lightsheet data, but also to continue the conversation about what Zeiss can do to better serve this community and help you accomplish your research goals. So my name is Monica Kirk. I'm a product marketing manager for software at Zeiss in North America. My role involves working closely with our global software development team to gather and incorporate feedback from researchers like you. And I'm grateful for this opportunity to be a part of this users group and collaborate in enhancing the software portfolio that we offer. With me today, I have colleagues who represent a nice cross-section of roles, so we can be sure to address any topics that come up in the discussions. Joining me from North America, we have Courtney Akatake, who's a Lightsheet product application specialist, and Renee Dalrymple, who's the head of life sciences and E&R business sector marketing. But you may have previously known her as our product marketing manager for Lightsheet systems in North America. Supporting us from Germany, we have Stefan Wagner-Conrad, who is a software architect and principal expert. Our speakers today will be Kevin O'Keefe from the US and Kirsten Elgas from Jena, Germany. Kirsty studied chemistry in Tübingen, Germany, with a focus on analytical and physical chemistry. During her PhD and subsequent postdoc positions, she investigated subcellular dynamics and morphology of live and fixed cells using confocal microscopy and correlative X-ray microscopy at the synchrotron. She then became an optical microscopy specialist for super resolution, FLIM, and light sheet microscopy in the microscopy facility at Monash University in Melbourne, Australia. After managing a node of this facility at the Hudson Institute of Medical Research for 3 years, she joined Zeiss first in Australia as a service and applications engineer, and now in Jena, Germany as application development engineer responsible for special 3D systems. Kevin studied biology in Albany, New York, with a focus on wound healing in response to mice. After leaving university at Albany, he became a 3D product application sales specialist for Zeiss in the greater New York area, covering all of our high-end light microscopy systems, including our Lattice Lightsheet system. He has since joined the new emerging business software sales team as senior software sales executive covering the East Coast, focusing on our Rebus solutions, which he'll discuss with you later today in the talk, and how they can benefit data handling, visualization, and analysis. So a quick overview of what we're going to talk about today. Kirsty is going to start off with some product highlights, some of the novel things that we have on the Zeiss Lattice Lightsheet that might be useful to you. And then Kevin will go into our Rebus software portfolio and how we're handling and analyzing our Lattice Lightsheet data at Zeiss. And then we'll turn it back over to Kirsty, who will talk about a Zeiss Rebus hackathon, some kind of software development news, exciting things on the forefront. And finally, we'll have some time for Q&A. If you want to type questions in the chat in the meantime, please feel free to do so, and we can address questions at the end. So without further ado, I will turn it over to Kirsty, I think.
Yes. Hi, everyone, and welcome to our presentation. And thanks again for having us and hosting us. We'll basically, as Monica said, we'll be presenting kind of the developments that Zeiss has made on this instrument, but we also seek your input. So in the end, we'll have a survey, and I'd ask you all to actually take part if possible. So that would be very interesting for us to get your feedback, and we value that very much. So I want to start off with just giving you a little bit of an overview of lattice lightsheet microscopy. Of course, you've all used lattice lightsheet microscopy, so I won't go into too much detail, but I will go then into more detail on what the Zeiss Lattice Lightsheet 7 has as unique features. So here, as most of you will probably know, or all of you, originally Eric Betzig published lattice lightsheet microscopy in 2014. Yeah, it was mostly back then everyone was blown away by the data he showed, because it was the first time we saw live cell data in such a high resolution and with such high temporal and spatial resolution. And for such a long period of time as well. But in his original prototype, basically, it was an upright system. So we had objectives dipping into the instrument, into the sample, and it was basically a system, home-built system that needed frequent alignment. So the challenge that actually Zeiss took on, and that Eric Betzig trusted Zeiss to actually make this into a commercial product, was to make his prototype into a system that can be used as an inverted system on standard sample carriers, and as easy to use as possible, so that it can sit in a facility, basically. And that basically had a lot of challenges, obviously, and I just want to go through some of the technical details that our development team came up with to solve and address those issues. The first, most obvious challenge, obviously, is if you want to image from below on an inverted system, you need to, but you still want standard sample carriers. You want to image through the cover slip at an angle. So basically, the design of the Lattice Lightsheet 7 objectives is displayed here. So you can place a sample that you would put on a high-end confocal or super resolution instrument as well, as long as it has a number 1.5 cover slip on the system. It has an illumination objective and a detection objective underneath. Detection at an angle of 30 degrees, illumination of 30 degrees, detection at 60 degrees. To be able to image through the cover slip and to compensate for refractive index mismatches, there's two main features. One is the meniscus lens that corrects for the air-water interface. So we basically have between the detection objective and the meniscus lens is an air gap, which makes the system extremely stable because the sample is thermally isolated. But it's yet another refractive index mismatch. And thank you. So we need to compensate for that. Part of that is being done by the meniscus lens. The second most important part, basically, and also very, very precisely manufactured parts, are the precision optics that we need to correct for imaging through the cover slip at an angle. So these are really to nanometer accuracy designed, highly complex polynomial surface. There are two plates that can be moved via software, basically. So the user just needs to pull a slider, but that corrects really very finely for the cover slip thickness, because as we know, it's 1.5, but it's still not perfect. So it can vary between 150 micron thickness and 190 micron thickness, and we need to be able to compensate for that. And that can only be done really with these high precision optics. So as you know, and this is not unique to Lattice Lightsheet, this is really unique, general to all Lattice Lightsheet technologies, because you're imaging at an angle. You have a skewed view of the raw data. This is basically displayed on the left-hand side here. So if you look at the raw data, this is a round T-cell, technically a round T-cell, but you see it looks very elongated and at an angle, so skewed. So we need to do the processing, and this is important for the future, for the next couple of slides that Kevin is going to present, and also what I'm going to present about the hackathon, to distinguish between processing and analysis. So here we are basically talking about processing. We really need to do a deskewing to be able to use the data afterwards, put it in the correct physical dimensions, to be able to analyze it properly. And you see after DSQ, basically the cell is round, and you would have done, all would have done that, because it's basically the same for all the lattice light sheet technologies, not just unique to the Zeiss solution. And then also for all lattice light sheets, basically the light sheets do come with side lobes, depending on how thin. The thinner the light sheet gets, the stronger the side lobes. So if you look at it in raw data, you get like mirror images, because you have these side lobes that are also exciting the sample. And we need to do a deconvolution to get rid of those mirror images. And that's, again, kind of the same for all the systems. In our system, we use a spatial light modulator to create the light sheets, and we give the user the choice to choose between 6 different light sheets. 3 of those have very minor side lobes, but they're also thicker. But basically, you can do the imaging without absolutely having to do a deconvolution, because you don't see the mirror images too strongly. But if you use the thinner light sheets, they're quite pronounced, the side lobes, and also the mirror images, as you can see in the left-hand image, basically are quite pronounced. And after deconvolution, because we know the PSF, the mirror images are gone. So all of this is basically processing, and it's absolutely required before we start to even think about any analysis. For the processing, the Zeiss offers a solution which we call direct processing, where you can basically hook up your lattice light sheet instrument to either a processing PC that Zeiss provides, or it can actually also be a server that you host, that your university or research institute hosts. And while you're acquiring the data is being streamed to the processing PC or the server and being processed as we acquire the data. So in the ideal case, your data is finished processing once you're done with the acquisition. That doesn't always work, especially if you do deconvolution, because it takes longer than the acquisition, can take longer than the acquisition. But at least you're using the time that you are while you're acquiring already to process. So this all is processing, but the acquisition at this point in time, the analysis at this point in time is basically still fully after the processing. And on the next slide, I've basically just invite you to have a look at those papers. It's from the Walter and Eliza Hall Institute in Melbourne. They do some impressive malaria research and some impressive analysis on the lattice light sheet. They've actually have a Geneva home build system. And they also got then a pre-serial lattice light sheet 7, and they've migrated pretty much all the or a lot of the projects, not all the projects, but a lot of the projects from the home build instrument to the lattice light sheet 7. Partly, for example, because it's much easier, because we offer a larger field of view to find cells of interest, especially if it's rare events. But they have done some very impressive data analysis, both on the data they recorded with the original home build Geneva instrument and the lattice light sheet 7 instrument. And at this point, I would hand over to Kevin, who is going to show you some of our analysis solutions, software solutions, before I take over again to present some of the ongoing developments.
Thank you, Kirsty. So Kirsty did a good introduction here about what Zeiss offers in terms of lattice and a little bit of background into lattice light sheet microscopy. When Zeiss released the lattice light sheet in December of 2020, we actually bought a controlling stock in a company, Arevis, and we've now fully integrated. And the idea behind that is to bring powerful image analysis solutions to the table, to the portfolio at Zeiss, not just to stop, like take an image and then stop at just the image, but to bring something tools that can get you results out of your answers. I mean, results out of your data. A few of the things that we have Arevis Cloud, which is a deep learning platform for training your own customized AI. We have Arevis Pro, which was formerly Arevis Vision 4D, where that's your desktop application where you're performing your analysis, you're setting up analysis pipelines, you're making like 3D rendering videos, things like this or taking screenshots for publication and doing your quantification. And then we have what's called the Arevis Hub, which was formerly known as the Arevis Vision Hub, in which case we can really scale up our analysis, do server 3D rendering, things like this. And what I want to point to you about the Arevis solutions are, yes, we work with the Zeiss ecosystem in terms of the hardware, but we also work with your existing microscope imaging platform. So everything that I'm going to talk about today in terms of the image analysis side of things, you can utilize your existing imaging platforms with. We work with, TIFFs. I think we have over 50 different file types we bring into the Arevis side of things. So I can share a link later on after the fact. And a lot of the Arevis solutions also integrate with third-party plugins such as Python or MATLAB, if that's of interest to you. But first, showing you the Arevis Cloud solution, this is where we take a subset of the data. Let's see, get this video playing here. Where we take a subset of your data, we upload it to the cloud, and we paint on it. So this works on partial annotations. So you're training the AI, your own custom deep learning model, by painting on it, annotating, providing ground truth. You click train. So you'll see where we click train in the upper right-hand corner. You don't need to have an AI expert on staff. Anybody can do it. And you download that and you use it for segmentation. So for something like maybe imaging conditions changing or intensity is changing when you're doing your Lattice Lightsheet imaging, you don't have to just set a threshold and say, okay, this will be good enough. You can train that AI to recognize maybe intensity changes or structural changes over time. So that's one of the benefits of deep learning in Lattice Lightsheet microscopy. Then once that model's trained, you download it and you bring it into the Arevis Pro platform, where we can answer kind of any research question from any modality, meaning, here we take Lattice Lightsheet data sets from different Lattice Lightsheet instruments. You can bring them in. We're always integrating new technologies into it. Kind of what I was just talking about is the AI we've been integrating. So you can train your own custom AI with something like Arevis Cloud, but also know we have integration with publicly trained and some publicly supported models, deep learning models such as CellPose and Stardust for segmentation of cells. We can work on data of any number of size and any number of results. And we have server-based solutions for when you want to take this and scale up. So looking at the Arevis Pro solution here, this is where we're going to do our 3D imaging. Here I like the 3D 4D analysis. So here I like to look at this little data set where we did segmentation with CellPose here, where we segmented out individual nuclei. They're all dense kind of within a structure. Then after that, in the software or the Arevis Pro, once you have your structures segmented, you can say, what do we want to do with them? Do we want to relate them to one another? Do we want to track them individually over time? Do we want to track lineages? And we'll talk a little bit about that later. And again, within the software, you have the ability to, if you have anyone in the lab or in the core, or you work with a collaborator that does their own Python scripting, you can make pre-processing, segmentation operators, and even post-processing operators, where you can really work with the open source community to create these different analysis steps and, really customize it to what you need, what you might want to do. Again, you don't have to be a Python expert to use this software. But if you have the ability to do it or know someone that can do it, you can really take advantage of it. And we've done some interesting studies in the past with people using Python scripting that we can share with you as well. So then once you have your analysis pipeline set up on the Arevis Pro, you can take it and move it to the Arevis Hub. So what is that? It's a web-based, it's a server solution that you log in via a web browser. You can have different user management rights, things like this. It can be on your local infrastructure if you have like servers in a closet that you can take advantage of. Of course, it has to be Windows for our software to work right now, but you can take advantage of that. We have customers that are deploying this on things like Microsoft Azure or AWS. But you use it to increase throughput, right? You have an analysis pipeline set up that you made in Arevis Pro. Now you're saying, I want to take my data sets and start parallelizing, meaning taking different data sets and saying, all right, you go to one computer and start your analysis, take the next data set, start on the next analysis. So it's a way to speed up your time from images to results. You can also use it for server 3D rendering. So if you guys are in a core, maybe you only have like one image analysis workstation. It's hard to render large data sets and be reliant on local workstations, things like this. So you have the ability to do some 3D rendering, data visualization on a server. You can use it to manage and schedule analysis jobs, and it's a centralized storage environment. As I mentioned, you can have different users that can see different folders, see different data sets, and share these things with one another. An example of maybe what one could do is, and Kirsty's going to talk a little bit about this in terms of the work that's been done in the hackathon, but let's say that you have a data set where you're collecting hundreds, thousands of images, hundreds of thousands of images, right? It's possible with something like the Lattice over a long period of time to generate data sets like that. If you have an analysis pipeline set up, just as a visualization here, if you had all of your time points kind of individually set up, you could take each time point, each volume, and send it to what we call an analysis worker, but think of it as another computer. So if you had 10 analysis workers, in an example here that we're showing, we're able to get a 10-fold increase in processing time. So reliant on one local workstation, pressing go would take about 9 hours to do the analysis. In this example we have here, where these were 2D, 1,000 images in 2D, again, just an example to kind of give you guys an idea, we're able to take it down to less than an hour. So to kind of paint the whole picture, what does the portfolio look like in terms of the software in general? Before we move on, you've got your local workstation that's right next to your Lattice Lightsheet instrument where you're collecting the data. If you're using the Zeiss Lattice Lightsheet, then you're using the Zen software to do your pre-processing, your deskewing, your deconvolution, should you choose to do it. And then you send that data over to Arevis Pro where you set up your analysis pipeline. If you're doing this sort of thing over and over again, and maybe you've got variability, you can train your own customized AI to help with that segmentation step. And when you want to take it and scale up, this is where you go to the Arevis Hub. But before we transfer over to talking about what we've done in the hackathon, I want to show you a little bit of what Zeiss has been working on. Is we have these published case studies that we can send you links to. One of the most recent ones that we published, it's called our From Images to Results, was actually imaging C. elegans on the Lattice Lightsheet. Now, what the researchers wanted to do in this example for this case study that we have is they wanted to track their cell lineages, and they wanted to take a look at centrosome dynamics over time and during divisions. So the researchers chose to use Lattice Lightsheet technology because it's sensitive, as we were talking about earlier, for doing long-term live cell imaging. And when you're following something like a centrosome, you're near isotropic in your XYZ, so you can follow those structures over time, not quite nicely in 3D. So what results did we see? We used deep learning to train a customized AI model to be able to detect where the cells are. So here we're looking in the Arevis Pro, a little bit of visualization here, where we're looking at every cell. We're following the specific lineages here. So you can see we've got cells in pink, we've got cells in red. You can follow all their lineages, and you can see on the right-hand side here the graphical representation of where we have these red dotted lines where we have division events, and then we're following individual cells over those periods in time to segment the different lineages. Then within each of those objects, we can look at centrosome dynamics. So what we're able to see, here's a video where we're looking at the centrosomes. They're segmented, they're pseudo-colored over time, so we can follow those centrosomes over time. We can see that when we're looking at cell division, that as cell division is increasing in these things over time, we're also seeing the centrosome number increase over time. But as we are looking at the global, oh, sorry, what is this doing here? Apologize for that. But as we looked at global centrosome intensity over time, our dotted lines are the division events that we're kind of looking at coming down in Y on the graph. So we see centrosome intensity is kind of cyclically mirroring the division events. But we did have an asynchronous division event where we see a division event and we don't see the centrosome intensity spike. So what you need to be able to measure that is actually following the specific lineages, looking at what is happening at every single lineage, every single cell lineage, to say what's happening to the centrosomes at each specific point in time. And we're able to do that with the tracking and lineage software in Arevis Pro. So here we can see all of our lineage, all of our different division events and all of our lineages. So we can look at the individual cells. And when we looked at them on an individual cell level, instead of looking at everything at a global level, we're able to actually see that if you look at time before division and then time after division, that centrosome intensity 10 minutes before division increases, then you get your division event and everything cyclically drops back down. And when we were looking at everything in terms of time points as a whole, in terms of centrosome intensity, we would not be able to get this if we weren't looking at the specific lineages themselves. So maybe I'll pass over to Kirsty here to talk a little bit about what we've done for the Zeiss Hackathon.
Unmute. Okay. Thanks, Kevin. Yes, at this point, so basically now we've shown a little bit what the system can do, what kind of processing is required, what kind of analysis is possible. But as I said before, at this point, maybe the processing is parallel to the acquisition, can be parallelized, but analysis is fully afterwards. So what we are trying to look at is at solutions, how to make that the first result basically accessible quicker. So one of the approaches we tried, and that's basically what Stefan's team demonstrated during the hackathon. That's also why Stefan is here as a support. So basically, instead of as we said we did before, we acquire one time point, we process it in parallel. Then the analysis is all happening afterwards, and we need to actually convert the ZI to this file afterwards, which can be up to 1 to 2 hours, but it can even be much longer if it's a big data set. So the idea was to actually not only do the processing in parallel, but to also do the analysis already in parallel. So directly after the data set is processed, we do analyze the first time point. So that can be a rendering or just a simple 3D rendering, or it can also be a fully blown analysis, like a volume analysis or surface change or something like that. So that was the idea. That's what we try to set up on an example experiment. And one of the things that we would like to get out of this is basically to ask your feedback whether you think this is a valuable approach. And that's why I'm also going to share a link and a QR code afterwards to a survey. So what we got from this first test basically was just simply cells that are undergoing mitosis. And if we first record the data and use direct processing to process the data, but then only afterwards start to analyze, the first time actually we do get a result, we do get to see some 3D rendered data is after 16 hours, basically fully after the recording and the processing has been done. With the what I'm going to call like a direct analysis approach during the hackathon, as a matter of fact, we do the acquisition, then the processing, then the analysis of time point 1. So the very first result we're getting to see, the very first 3D rendering is after 5 to 10 minutes. So that is basically, in this use case, it's 100 times faster to get to the first result. Obviously, it will still take until the end of the experiment or longer to get the full result, especially if you want to look at tracking or something like that. But at least you already get a very quick first impression of what your data looks in 3D, and maybe also the first time points where something happens or not. So that's the idea behind it, as it was set up in the hackathon, but that doesn't mean that's kind of a final implementation. So what I should also say, I'm talking about a hackathon because it's really something that we are trying. that we are working on, that we are looking into, but it's not a final product yet. That's also the reason why we are actually asking for your feedback, for the community's feedback, to know whether you guys think this is really something that is worthwhile, or should we rather look at completely different options. Anyway, so the way that it's set up during the hackathon is basically we have one, the acquisition PC that is running the experiment, kind of missing my data set here. Then we have the processing PC. Yeah, that should be running. Anyway, so it's mitotic cells, basically. It's a standard LLC PK1 cell line with microtubules and histones labeled with cherry and microtubules stained with GFP. So we use this quite often to look at just at mitosis and look at how gentle a system is and whether cells go through mitosis. So that's what you should be seeing here on the acquisition PC. The storage PC is basically in parallel to the acquisition, doing the processing, as in doing the deskewing, potentially doing a deconvolution, basically get the data set in the correct format to make analysis correct and worthwhile. Then we also have an analysis PC that does, once the first time point is processed, does the analysis, here in this case using the Arevis Vision Hub, does the analysis in parallel while processing and acquisition is running. And if you want, in this case, we even set it up with a fourth PC that is a visualization PC that then you can use directly to look at the already processed time points and already analyzed time points. So here we can just simply see a 3D rendering. But as I said, that's not limited to 3D rendering. Kevin showed some of the stuff you can do with Arevis. The malaria data that I was mentioning before is also looking at, for example, surface area and pinching off of the membrane and so on. So there's a lot of obviously image analysis capabilities behind Arevis. That being said, doesn't necessarily need to be Arevis, but that's how we set it up. And yeah, as I said, not a product yet, but it's something that we are looking at. So we really would appreciate your feedback. On the last slide, I've actually put in a QR code and we'll also put the link in the chat. You can answer the survey either straight away or you can do it in your own time afterwards. But it's basically going to ask you a little bit about, for example, first of all, which Lattice Lightsheet system you're using, but also what do you currently use to store your data, what format do you store it in, and so on. Do you store the raw data, other data, and most importantly for us, obviously also feedback on whether you think this direct analysis approach is actually valuable for you and your research or not. So I would highly appreciate your feedback. Thank everyone for the attention, and I think we have plenty of time left for questions. So the whole team is available for questions. So let's open the Q&A and take it from there.
Excellent. Thanks so much, guys. That was really, really exciting. Yeah, I think I have a ton of questions, but I'm going to leave hopefully some of the other members. I'll fill some silence if nobody else wants to. But yeah, and you can certainly come off of mute, come on camera, ask a question if you'd like. If you want to put it in chat, do it that way. Yeah, however you'd like to do it. Yeah, I'll start with a simple question.
Okay.
So in your hackathon, that was super interesting. So, you know, your 5 to 10 minutes, is that kind of like a little bit longer than what it actually is? Because I mean, obviously you have to acquire the data and then do something, right? So the time to do the processing and visualization, like if you were to put an actual time on that bit's probably a little bit shorter. Is that correct?
Okay, I'm not even sure I got the questions on
So timeline, maybe if we if you had that slide, that might make it a little easier.
Yes.
I'm just curious how quick it actually, because it seems like you're probably doing it a little quicker.
You mean if you can get to the result quicker than 5 to 10 minutes?
Oh, no, I'm just asking if you actually are getting, like, to me, it seems like the time bar there, we have it from the start of the acquisition to the analysis of T1.
Yeah.
From a fair point, it seems like you're green to yellow. I was just curious what the green to yellow time is.
Right, yeah, okay. The green to yellow time will probably depend also mostly on what kind of processing parameters you set. If you just do a DSkew, it's quicker, obviously, than if you do a deconvolution with, I don't know, 20, 30 iterations. So it's very, very, very unique to what kind of processing you actually do. With the quickest, with the DSkew, you would probably be quicker than the 5 to 10 minutes.
Yep.
But if you do a deconvolution with multiple iterations, it will generally take a bit longer than the acquisition itself.
And yeah, and DSkew only is can be pretty fast, right? And it depends also on the size of the volume. So if you acquire a very long lattice light sheet volume, right? So it takes also more time. But in general, lattice light sheet is very quick in acquiring volumes. So
are you also rotating the data? It seems like still the DSkew, like if you look at the YZ projection on a DSkew data, that's still at an angle with respect to the cover slip. Are you deskewing and rotating? Is that considered one operation, or are you just doing a deskew and leaving the cover slip like the cell alpha?
Yeah, we have different modes, and I think one of the modes is the cover slip transformation. But I'm not sure maybe what Kirsty showed beforehand.
I think we showed directly with the cover slip transformation. So as the in Zen, there is actually two methods to do the deconvolution. Sorry, the deskewing. One is just the deskewing where it corrects for the skewedness, but you still look at the sample from the angled view. The other one is really what we call deskew plus cover glass transformation. And the cover glass transformation does the coordinate transformation to get your sample. In the standard lattice light sheet sample would maybe be a Tiran cell cultures, to get that into the standard inverted view that you're used to from a confocal. So some people really that they are used to confocal images, and they know how their cells look like from inverted view from below or above, but they are not used at looking at cells at an angle. So that's why we offer the option to do a cover glass transformation straight away, which gives you the inverted view and the more familiar view, depending on your acquisition and the sample that you have, on your acquisition settings and the sample that you have. It may also not be a good idea to do the cover slip transformation, especially for something like a spheroid or Drosophila embryo, or that is round anyway. You don't care too much about what angle you look at. But it may be it may be quicker to actually just do a deskew, and more so safe on file size, depending on your experiment parameters, to just do a deskew.
Looks like we have a question in the chat. I can either read it, or Praveen, if you'd like to ask your question.
Yeah, go for that. Can you hear me?
Yes.
Yes.
Great. I just have two questions. So from what I understand, the Arriva's file format, SIS, is currently not bioformats compatible. Please correct me if I'm wrong. Is there a plan in the future, to make it bioformats compatible? And the second question that I had is there a GitHub page that I can check out for the hackathon that was organized?
Okay, so good. Maybe I can take this. So so there's actually no plan yet to add the SIS format to the bioformats library. It has been discussed internally, but we don't have a definite decision on that yet. So it's not available. That's correct. We're still kind of clarifying internally how to go about there. And for the second question, so parts of what we did for the hackathon is changing parts in Zen, implementing some intermediate utilities, or changing the Aribis hub. And this is kind of all internal code, and it's not something that that's kind of possible to put in a GitHub repo for us.
So I had a question. You had mentioned that most size limits can be accommodated, but I wonder if you have kind of a practical limit of a data set that could be loaded and analyzed in a reasonable amount of time. And sort of a second part of that is, what kind of computer do you need? Like, what are the key components, and maybe a ballpark of how much that would cost as well?
Yeah, so again, let me take the second question first, which is data sizes, computer builds, things like this. We have a web page that makes recommendations on the PC for what we recommend as per data sizes. So we have a couple of ranges of data set sizes, and then if you're doing those larger sizes, we have something you might focus on for Lattice if you're making big data sets, is like 500 GB and above. So we have recommended PC builds. In terms of prices, you can buy a PC from us, but you don't have to. So if you have your own analysis PC, as long as we have the requirements for what you need at bare minimum for that, and then we have recommendations on what you want to put into it as well. So no need to come directly from us. I think Monica's pasting some things in the chat. Monica, did you put the PC build page in there? If not, I can. Okay,
No,
you put this.
I was getting there. Sorry.
Okay, so Monica will put that in there as well. And then in terms of data size and how long it takes to work with these sorts of things, do I have a good idea? I think a lot of kind of what Kirsty alluded to in the hackathon and the old way of doing things is a lot of that time for some of these data sets can just be even the conversion to the SIS file format type, right? So that's where you're going to spend a lot of your time. Then in terms of the analysis, I think it depends upon data size. So I don't want to go into and what you're looking to do, because some operations take longer than others, meaning if you want to do a basic 3D thresholding or maybe like a blob detection, things like this. Stefan, did you want to add something in for the times and data sizes there maybe?
Yeah, I wanted to add something with respect to the conversion to SIS. So we are working with the team that takes care of Aribis Pro to make ZZI a kind of a first-class citizen also in Aribis Pro. So to circumvent unnecessary conversions of terabytes of data, just for the sake of loading them in Aribis Pro. This is something that is in the works.
From a kind of a technical standpoint, I noticed you had 3 lasers listed. 405 is not listed. Is there ever going to be a 405 option, or is that just a limitation of like the SLM? Because I know it's really tough.
So there was the decision was made against 405 based on a technical and an application reason. Obviously, 405 nanometer light is quite damaging to cells. Mm-hmm. Especially for lifestyle imaging, it's anyway not that suitable. But the main reason for us basically was really the technical solution, and that's the technical reason, which is already what you said, is really the SLM. So as you know, there's 2 types of SLMs, and the Eric Betzig system uses the I think the ferromagnetic one, which is not as efficient, but can deal with 405 nanometer light. While we use a different one, the pneumatic one, and that is easily damaged by 405 nanometer light, but is a lot more efficient. So I don't have the number on top of my head, but I think 80–90%, something like that. Anyway, it's definitely much higher, which means for us that we can have low laser power, low power lasers, which means that it's a laser safety class 1 system rather than something like the original prototype, Eric Betzig Janelia 3i, where you need really 500 milliwatts to 1 watt lasers to get enough sample to get enough light to the sample. So that was the technical reason, and in combination with the application reason that 4 or 5 nanometer light is anyway too damaging for cells. We are not looking at implementing a 4 or 5 nanometer laser. If anything, you would rather look at longer wavelength. That's not in the near future, but it's certainly something that has come up and has been discussed.
I had a question about the Arrivis Hub. Just what kind of support do you offer to customers for setting that up and maintaining it? Is it something fairly straightforward, or does it take some computing expertise?
It takes some computing expertise, and it is something that we do bring to the table for you. So in covering North America right now, not on the call, we did just hire an IT expert who does will do the installation for you, will help with the technical side of things, can talk to your IT if you guys have that IT in-house. So we are building those resources, and we have some resources right now in terms of that. Like locally in the US and North America. And again, we do have a team on the Arrivis team, a full development team that supports this as well.
Looks like there might be one other question that hasn't been addressed. Do you have a ground truth data to test and validate AI algorithms? Not sure where that came from, but it's in the notes.
That was me.
Oh, was it?
Oh,
Okay.
Yeah.
Sorry,
So
I'm
just-
looking in the chat for
Yeah,
it.
If you have anything to test and validate the AI algorithms.
Yeah, so when you are you talking about models you train with Arrivis Cloud specifically?
Yeah, I guess the AI training and so on, like, is there a way to
Right.
validate that the analysis is accurate?
Yep. So from what I have, this is from my understanding, and if someone has a more technical understanding, they can maybe lean in on the question as well. But for practical use for users, what we typically recommend for them to do is take regions of interest or different images that are all variable, right? Add them onto Arrivis Cloud in terms of your training data set, and then you're performing the kind of iterative process of training and validating that model. Within the way that the model is trained itself, when you add the annotations in, and I think I could point you to maybe the people on the back end who can talk to it a little bit more in depth. But what you do is you're performing annotations to get started. We typically recommend like 100 or 150 annotations to start with a model, right? In terms of objects of interest. So it's not necessarily a number of images, but it's objects of interest. And during the training process, it's my understanding that half of the annotations that you've done are taken to create the model, and then half are used for some sort of validation of that model inside the model itself. But validating it by eye, making sure that it's getting the right structures of interest, is something that we recommend to the end users to do, to check and kind of go through an iterative process, right?
Thank you.
You're welcome.
I have one more question. If nobody else is, I'm going to ask all the questions. I don't care. So, is the water meniscus lens? Do you have to continually fill that, or I'm trying to understand it in terms of like the application? Is that filled? And then the second half of that question is, in the little schematic, and this is probably just the drawing, but it looked as though the focus of both the light sheet and the collection was at the focus of that lens rather than inside the sample. Is that just a drawing to make it easier, or seemed a little odd?
Yeah, the latter one is probably just not accurate enough
Sure.
drawn, accurately enough drawn. To the first question, basically the meniscus lens needs to be filled with water. Yes, it's basically your water immersion as you would put on a for example, a water objective. We have two ways of doing it. So if you do a rather short-term experiment for an hour or less than an hour, technically you can just use a pipette and drop some water on the lens, and you will be able to image for an hour without problems. But obviously you're having incubation conditions usually with live cells at 37 degrees. Water will evaporate over time. So if you want to image for hours or days, it's actually not possible to keep refilling manually. So we have auto immersion tubing basically going into the meniscus lens. So there's one tube that's and a reservoir is underneath the instrument, or in the instrument technically underneath the objectives. At the front door, you can open the front door and access the water reservoir. And then you basically have pumps that pump water from the reservoir into the meniscus lens. And you can control this in the software. So it needs a little bit of expertise to set it up in terms of it's very dependent on the local environment in your lab, depending on the temperature and so on. It will take different, will need different timings. So it will depend also on the humidity that you set up in your incubation, how often you do a refill. But ideally, once you've set it up, and you also need to prime the system obviously properly, otherwise there will be bubbles that interfere with your imaging. That's why it's also one of the reasons if you do a short-term experiment, you just drop a drop of water on because you don't want to go through the trouble of priming everything properly. But if you do all of that, set it up properly, basically end up with refill every, let's say, 20-30 minutes, automatic refill of the auto immersion. That's about as much as will evaporate. So we only add very little water each time to also not have the sample move up massively for each iteration, for each pump stroke. So it will be, I think, good times usually are about 20 to 30 minutes, depending on the humidity that you set up in your incubation. And then, yeah, the system will refill basically for as long as you want it to refill.
Thank you. Any other burning questions from anybody? Looks like we had a comment from Andrew, which, yeah, so six lasers, that's exciting on there. Very cool.
And I have already had some responses to the survey, so thank you to those ones, and it seems to have worked. So I'm looking forward to reading through the responses.
I think we're coming up on our hour, so I think that worked out really, really well. Thank you guys so much for presenting and taking the time to really be very engaging. I really enjoyed this, so thank you so much.
Thank you for having us.
Yep,
Thanks for inviting us, and please do reach out. I put some links if you scroll up in the chat. There are links to reach out to Kevin and Courtney if you have software or hardware questions, so we can address your needs directly. We really appreciate taking the time and are open to your feedback. And Monica, I'm going to talk to, I think you said Chase is working on your demo, so we'll get in touch with him too.
Great. Thank you, everybody.
Thank you.
Thank you, everybody.
Next one is November the 7th. Vanessa dropped that in the chat. So look forward to seeing you then.